The root knot nematodes of the Meloidogyne family are obligate parasites of several thousand plants, grass, and trees, cultural and wild plants. The Columbia root knot nematode, Meloidogyne enterolobii, and the false Columbia root knot nematode, Meloidogyne phalanx, are parasitic in monocotyledonous and dicotyledonous plants, including several crops such as potatoes, carrots, and tomatoes. The gall nematodes are widespread across the world and are detected on almost all continents. These dangerous pests cause damage up to 5% of worldwide yield of cultural crops. Many of them inhabit tropical and subtropical regions and are found in the protected ground only under moderate climate conditions. But there are also open ground nematodes that cause extensive damage to agricultural and ornamental plants in temperate latitudes, including the nematodes that can enter our country from Europe, Meloidogyne enterolobii and Meloidogyne phalanx. These are very harmful nematodes. The root knot nematodes are small round worms. Males are of a vermicular shape of up to 2 millimeters. Females are bloated, oval, jug-like, pear-like shape of less than 1 millimeter, white or pearl color. Nematodes penetrate the roots of the plants, causing acromentation of tissue, resulting in the formation of bumps, neoplasms, and so-called galls. As a result, the suction capacity of the root system is reduced. Over time, the plant weakened by the nematode dies back. The root knot nematode is a root parasite. It affects the roots of the plant. The plant runs out of nutrients, and the crop is lost. For example, in vegetable crops, up to 80% on cucumbers and 20 to 30% on tomatoes. The open ground quarantine nematode, Meloidogyne enterolobii, causes great damage to grains and potatoes. At the initial stage of plant infestation, the bodies of the female nematodes may be on the outside, and the head of the parasite will be immersed in the plant tissue. Later, as the root tissue grows, only a small part of the pest can be outside. Living nematodes are almost indistinguishable by color from the root tissue. The best result in the extraction of nematodes is to split the root along the fibers by a microscopic needle. In this case, the bodies of the females remain whole. That's how the egg-shaped bags become visible on the surface of the roots. The eggs are oblong, rounded, contain a rolled-up larvae. The nematode can also enter our territory with regulated articles where non-quarantined species are frequently present. The quarantine root knot nematode species have not been detected. Therefore, it is important to distinguish non-quarantine species from quarantine species. At first, the plants affected by nematodes show symptoms of nutrient deficiency and moisture, and the plants appear to be depopulated. The primary external manifestation of the infestation by root knot nematode is galls on the ends of the roots. If an examination of root system identifies such structures, further diagnostics are carried out using specialized techniques developed at the All-Russian Plant Quarantine Center. The Columbia root knot nematode is the most dangerous for our country, but there are virtually no pest-resistant varieties and highly effective nematicides. The root knot nematode is quite difficult to control. In general, crop rotation is used to control the nematode in open ground, but the Columbia nematode affects grains besides potatoes, so the rotation of potato and grains is not applicable in this case. In America, they rotate potatoes with turnip and radish. In the greenhouses, heat treatment of soil or full replacement of the substrate is helpful. Biological methods of controlling root knot nematodes are to improve the soil with the number of organisms feeding on the invasive nematode larvae. These can be predatory mites, predatory mushrooms, or predatory nematodes specifically propagated and put into the soil. For this, organic fertilizers, sugar, or waste can be put into the soil. This organic material helps to develop a fair amount of saprophytes, followed by the reproduction of natural predators.